A very good morning. A very good morning, everyone, and welcome to the final day of Fungal Awareness Week 2025. Today, we will focus on practical aspects through a demonstration and an interactive quiz. I am Dr. Sharlini Devi Gunasegaran from Hospital Sultana Aminah Johor Bahru.
And I am Dr. Alia binti Abdul Rahman, medical microbiologist from Hospital Sultana Aminah Johor Bahru as well. Uh, I have the pleasure of introducing the CV of the amazing Dr. Tan Zui Ting, our speaker for today. Dr. Tan Zui Ting is a research officer at the Infectious Disease Research Center, Institute for Medical Research. Her expertise lies in molecular biology, infectious disease, and drug discovery, with a focus on antifungal resistance. She is currently leading and collaborating on multiple research projects, including antifungal susceptibility of Candida, Aspergillus, Fusarium, and Trichophyton species, as well as COVID-19 diagnostics and vaccine development. She has served as principal investigator on several Ministry of Health funded projects and has presented her findings at both national and international conferences. Dr. Tan has authored multiple publications in high-impact journals, including recent work on antifungal resistance in Aspergillus fumigatus, Cladophyllophora bohpii, and Thalaromyces monophyte. She also contributes to postgraduate teaching and training in medical mycology. With her strong research background, she represents the next generation of leaders in fungal diagnostics and resistance surveillance.
It is my pleasure to introduce the experienced Dr. Siti Hawa Hamzah, a medical microbiologist. She currently acts as head of microbiology unit at Hospital Seberang, Jaya. She also serves as the state microbiologist for Penang since 2010, providing leadership across the state's laboratories. Dr. Siti Hawa obtained her MD and Master of Pathology in Medical Microbiology from University Science Malaysia and further trained in medical mycology at PGI-MER, India. Her special interests include antimicrobial stewardship and medical mycology. She's an active member of professional societies including MSIDC, MMIG, ISHAM, and MICE.
Attendance link will be provided at the end of the webinar, together with a feedback form. CPD points and e-certificate will be given daily, and a total of 8 points will be given if you have attended all 3 sessions.
Okay, gentle reminder as well, the Q&A session will be at the end of the demonstration, and there will also be an interactive quiz. So if you do have any questions, you can type the questions in the Q&A box and raise your hands later if you wish to speak. Okay, so we will now proceed with the demonstration by Dr. Tan Shui Ting and her team, covering tape mount method, tease mount method, and fungal slide method. Dr. Tan, the floor is yours.
Thank you, Dr. Shalini and Dr. Alia for the kind introductions. So welcome. Good morning, everyone. Welcome to the Fungal Awareness Week organized by MSIDC and MMIG. So today is the last topic of the week. So what you can see on the screen now is an intricate architecture of a common mold Aspergillus. So to the naked eyes, it might look like a fuzzy, green, or black spot on a forgotten food. However, when it's under the microscope, it reveals itself as a complex and a surprisingly elegant organism. So the hidden world is what we are going to discover today. Ladies and gentlemen, my name is Tan Shui Ting. So I'm a research officer in IMR. My work delves into the cutting edge of the fungal resistance, from the antifungal susceptibility testing of mold to the Aspergillus and Candida resistant genes detection. So, but it's our upcoming published paper on the Trichophyton indothene that highlights a critical truth that even though how advanced the research you build, the fundamental of accurate identification of fungus is still very relevant. But of course, as we all know, we can't identify the fungus based on the color alone. The true identity of the fungus is written in its microscopic details. So to read the text, we need to have the specific tools and reagents. So in this session, we will dive into 3 fundamental techniques that allows us to prepare the sample, the fungal sample. So these are tap mount, teeth mount, and slide culture. So the goal of the microscopic examination is allows us to see the fungal structures clearly, intact, and in their natural arrangement. So to unlock this view, we need to have specific tools and reagents. So let's see what we need on the bench to get started. So these are the universal tools and reagents that we need for any of the method that we are discussing today. So the basic one is the microscope slides and cover slips. A set of sterile tools such as tweezers, teasing needles, and inoculating loops to manipulate your samples. So lactophenol cotton blue or LPCB stand. So it's very important for us to work with the mold inside the biosafety cabinet. So it's important to avoid the spore inhalation. So the most critical item for all this technique is the LPCB stand. It's not just a simple tool. It's a multi-tool reagent. All right. So because each component has its own job, for example, lactic acid, so it preserves the structure and maintains the structural integrity of the fungus and prevents it from distortion. So phenol, it can kill the fungus, so it is crucial for our safety. So the cotton blue, it is what provides the contrast because it selectively stains the chitin in the cell walls and make those beautiful and intricate architecture visible against the background. So glycerol, it prevents the sample from drying. So with all these tools and equipments, microscope, biosafety cabinet are ready. So now we move to the easiest and simplest technique. So tap mount. So sometimes people call tap mount as a cellophane tap mount or Scotch tap mount. So the general principle of the tap mount is we use a short length of a clear and adhesive tap. We gently press it over the surface of the fungal colony. So when we remove and lift the tap, so the fungus will adhere on the tap. So then we transfer the tap onto the slide. So we can proceed for microscopic viewing. So this mounting preserve its natural or native arrangement without any disturb for the microscopic analysis. So let's say if you want to have a rapid or preliminary look on what kind of spore of the fungus is producing, so maybe this could be your first choice. How does it done? So let's walk through step by step. First, you place a drop of LPCB on a clean microscope slide. Then we gently press it onto the surface of the fungal colony. Then we carefully lift the tap. Then we place the tap, the sticky side down, onto the drop of LPCB on the slide and avoid any bubbles. Then it's ready to examine under the microscope. So this is the demonstration video. So in this video, we'll show you how to subculture the mold, and then the staff also will show you how to do the tap mount. So she also has briefly mentioned the step in Bahasa Malaysia. Hope you guys will enjoy this video. So now we have 2 types. The first one is the subculture for invasive sensor. This one they collect the spore. Yes, they fully into lysis buffer. Are ready for PCR. For the non-invasive sample, this is what we do.
ataupun
Okay, this is a non-invasive type. So for tap mount, we use the Scotch tap mount. So we check the growth daily. So first we label. Then we cut a short length of a fungi type. Yes, we avoid from touching the sticky side. And then we press the sticky side onto the colony, the surface. Then we transfer the tap onto the slide. Okay, so that is the demonstration for the tap mount. So the main advantage of the tap mount is it allows us to quickly capture an undisturbed snapshot of a fungus surface features, right? For example, we can see the microscope, we can directly see this, the canis, okay, clearly. But its main disadvantage is it does not show the internal or submerged structures. But this is the true picture that I took when I first learned the tap mount. Is actually the aspergillus, and you see the number of the spore is too many. It has actually covered the vesicle, the phylates and the medulla. So we cannot examine what is this species of this aspergillus. So in order to have a more comprehensive view, we will move to another method. So this is teeth mount, or sometimes people call it as needle mount. So now we imagine tap mount is like a photographer. Why? Because we take the pictures, we take the picture of its surface, the fungal surface, but teeth mount, by contrast, is being like a surgeon. Why? Because the core principle of the teeth mount is the mechanical dissection. So the general principle is we select a small part of a fresh fungal colony and we gently separate it by teasing, pulling or tearing into a tiny and flat fragments to spread out the structures for microscopic observation. So this mounting is ideal for the fungus that is too dense with the spore, like Aspergillus, or maybe it's embedded in the agar, or maybe it's powdery or very cottony. Yes, and sometimes we use this for the dimorphic fungus. Okay, so step by step. So the first one, we put 1 drop of LPCB stain on a clean glass slide. Then followed by, we pick up a small portion of the fungal colony, with cutting through the area and vegetative mycelium. We place the fungal fragment into the LPCB drop. So we gently tease the fungal fragment apart. So by this process, actually many spores will be detached and floated off. So your core principle of the tease mount is you fragment the core, you fragment the clump into the individual hyphae and spore. So after that, we apply a cover slip and examine. So now we continue watching the tease mount. Okay. 或者叫的 LPCV. And this is Aspergillus niger. We take a small piece, yes, including its agar. Then we tease it. Yes, then we place a cover slip on top. So you are spread. Yes. So in this step, we doesn't need the tap mount. We doesn't need the tap mount. Yes, we doesn't need the paper, the cellophane tab. Right. So let's continue. So the main advantage is still considered rapid, and it allows us to have immediate observation of fungal structures, including hyphae. But the main disadvantage is when the time we separate the fungal culture, we tend to damage the delicate features, especially the arrangement of the spore change and the hyphae, and it can make the accurate identification challenging. So now we need another method that preserve the beautiful fungal architectures perfectly, so without disturbing them. So this leads us to slide culture. So sometimes people also call it as microculture, right? So now we imagine again, the photographer is the tap mount, all right? Surgeon is the teeth mount. So what about slide culture? Right. So slide culture is someone like an architect because it allows the fungus because it allows the fungus to grow directly onto your microscopic slide. All right. So the principle of the slide culture is very brilliant. Instead of sample the mature colony, it let the fungus culture and grow directly onto your microscope slide, and it allows it to grow in its natural form in 2 dimensions under a cover slip. It's like an architect building a mini observatory and gives the fungus a window to grow and grow directly onto your microscope slide. So it led us to view the most delicate and most beautiful architecture without disturbing them. So now, building a mini observatory requires careful setup. So we start by preparing a sterile microscope slide. Then we cut a small block of agar from a fresh plate. For example, in this case, we use PDA to enhance the sporulation of the mold. Then we inoculate the sides of the agar block with the fungus. Now we have one agar block like this, square. Then we inoculate the fungus into 4 sides of the agar. So this agar block serves as the nutrient source for the agar, for the fungus to grow. Then after that, we place a cover slip on top of the agar block. Place on this. Then we transfer the whole slides into a petri dish with a rod or stick as a support. Then after that, in our lab, we place a sterile water dampened cotton ball in the dish to maintain the humidity. So in our lab, we use a sterile water dampened cotton ball and we put it aside here. So it's up to your lab. Some of the lab, they place a sterile water dampened filter paper. So as long as it serves the same purpose, then it's all right. then it follows by incubating. So now the waiting game begins. You have to check the growth daily. So let's say if you are satisfied with the growth, then it's the time for you to harvest. For example, after 2 to 7 days of incubation is depends on what is your isolate. Then you can see actually the agar block here, 4 sides of the agar block are grown with the fungus. So now you carefully remove the cover slip from the agar block, carefully, slowly, and vertically like this, not smearing the fungus, right? Then after that, we place the cover slip. So if, let's say, if you have removed the cover slip, you see the underside of your cover slip actually is still full of the fungus. Then that cover slip can be mounted with a drop of LPCB on the new slide. Then you discard this agar block. But wait, it's not done yet. After discarding, so we can mount this original slide, right? With a new cover slip and LPCB, something like this. Then we examine the slides under the microscope. So now we continue with the video. Okay. TDA agar, we kept a small blood. We transfer this agar block onto a cover slip, onto a slide. Both sides. So we put a cover slip on top. So this stick will serve as the support. Break it half. Break it half. We put into the petri dish and serve as a support for the slide. Yes, we transfer the slide onto this formation. We put it aside here. So we incubate it. So this is step 1. So proceed to part 2. After incubation, you see 4 of the slides are full with the fungus. We put another gel of FPCB. Then we transfer the slit on top of the agar and put it here. Then we discard the agar. Then we mount this original site. So now we have created 2 slides. We created 2 slides from a single colony. So it let us to have a multiple views for the fungal growth. So let's continue.
Agar kan yang kosong kan, okay. Cover slip kita atas tadi tu kita letak dekat sini, so kita akan nampak bahagian top. Kita buat agar tu, kita letak standing lagi satu, okay. Kita ambil cover slip baru, kita letak kat sebelah kiri ni. So macam ni kejadian di bahagian atas, top, bahagian bawah bahagian. Dan nampak atas dan bawah.
All right. So this is the site culture. Right. Okay. So the problem is which technique should we choose? So perhaps this table can give you a basic guidelines. For example, tap mount is primarily used for the native arrangement for spores and fruiting bodies. And process is very fast, simple, and it preserves the morphology without disturbing the structures. However, it's only superficial and sometimes people can messy with the tap. T-mount is primarily used for us to see the individual hyphae, conidia, spores. It is also quick, easy, and suitable for mold with dense mycelium. However, it can be disruptive to the conidia arrangement, hyphae, and its position. The slide culture is primarily used for us to see the undisturbed and complete morphology of all fungal structures. It gives us a perfect intact structures. For example, we can see very intact and its delicate structures of the conidiophore, spore change, and hyphae of the fungus like Aspergillus and Penicillium because it preserves every minute details for the conformative diagnosis. However, it needs a long time for the incubation and setup. It's time consuming and limited nutrients because we only provide a small block of the agar. So summary is you choose the technique based on what you are looking at and what do you want to see. So for example, if doctor asks what is on the surface or how do the spores look like on the surface, then perhaps you can first choose the tap mount. Then after that, so let's say if you want to see the hyphae structures or large or tough reproductive bodies, then maybe you can choose T-mount. Or what is the perfect or undisturbed form of this fungus for a definitive identification? Then maybe we can proceed straight away to slide culture. However, things doesn't always go perfect. So we always have some problems. Then this is our time to solve this problem. So you see, this is the real picture that I took when I first learned the tap mount. So you can't see anything except the spore, right? Because the site is full with or dense with the spores. So for heavily sporulating cultures, tap mount might be too dense. Perhaps you can try T-mount or slide culture. Or no sporulation. we can straight away try slide culture because it can often induce sporulation in reactant fungi. Or sometimes after you cover the slit, you will see a lot of bubbles. So my experience is I put another drop of LPCB, just one little drop, and place a second cover slip on top and press gently to slide the bubbles out. It's up to your lab. Maybe you have different technique that you can share with us. But the perfect way is we tilt the cover slip about 45 degrees to the slide, and we slowly lower down it, right? But some of the lab, they briefly flame the slide to remove the bubble. Then there is other lab that incubate the slide. After the LPCB, they incubate the slide into the vacuum chamber. So it's their way to remove the bubbles. Okay. So if let's say if nothing visible, we can start with the low magnification to locate the areas of interest, then only we move to higher power, like 40 or 100 for detailed microscopic examination. So that's my sharing. Thank you very much. So
Thank you very much, Dr. Tan and team, for the impressive demonstration.
We will now open the floor for any questions regarding the video demonstration and the fungal techniques. If you have any questions, you can raise your hand and then we will unmute you, or you can type it in the Q&A or chat box. I have a question for Dr. Tan. If, say, the center does not have PDA, potato dextrose agar, what other agar is suitable for the slide culture?
slide culture. Sometimes people use communal agar, but the best is the PDA because it enhances sporulation. Yes. Commune, sometimes people use it for the yeast, but for PDA, for the mold, people use PDA, yes. Because we want the sporulation. For example, if let's say you don't have PDA, you don't have commune, or let's say it's still no growth of these two types of agar, then we have to straight away proceed to molecular. For example, you have to do PCR on its ITS reagent. Yes. Mm. Yes.
So if there are no more questions, so we shall proceed. So to
Yes.
wrap up Fungal Awareness Week 2025, we now invite all participants to join in our interactive quiz session. Please
Yes.
participate actively and have fun at the same time.
So the participant here can scan the QR and we have the link, right? Dr. Shalini and Dr. Alia, we have the link. Can we share the link into the chat box also?
Okay, we will do that.
Mm-mm. So can everyone also can log in to answer the quiz, right? The QR and the link are working. So this is the quiz.
Dr. Tan, can we give participants about 10 to 15 minutes for this?
Yeah, sure. So everyone also can enter, right? Can access the link and the QR code, correct?
Yes. The link is already in the chat box. So
Yes.
people can click on that.
Yeah, so this is the fungal identification quiz with the clinical and microscopy insights. So we have provide some brief clinical situation. For example, the first one case of a fungal meningitis in immunocompetent patient. So we also provide the the morphology and its LPCBs or Indian ink stain like this.
We will stop at 11 a.m.
Okay, 2 more minutes, people. Okay, it's already 11 o'clock. I hope everybody had fun trying to answer that. The moderators definitely did have fun. Okay, just don't forget to fill up the attendance for ESET and CPD points, which we will blast in the chat box and later on. Feedback for this Fungal Awareness Week will also be included in the attendance link. Please give your honest opinion in order for us to further improve for next year's event. And with that, I would like to invite Dr. Siti Hawa to give an explanation on the quiz and the answers. Okay, thank you.
Assalamualaikum and salam sejahtera. Good morning, everyone. Thank you, moderators, Dr. Shahina and Dr. Alia. And thank you also to Dr. Tan for a very good presentation on the technique. So now, if you think the life is hard, so I can tell you now that mycology is also hard. But actually, everything is hard before it becomes easy. Okay, so we go to the first slide. Okay, first case. Okay, this is actually a case of fungal meningitis in an immunocompetent patient. So the keywords here is actually an immunocompetent patient. Okay, so we can see that there is encapsulated yeast. Okay, in an Indian ink, and the most likely organism would be Cryptococcus gatii. Okay, so we are very familiar with Cryptococcus neoformans, but there are also Cryptococcus gatii. So this gatii is actually an environmental basidiomycetes yeast, and then it is associated with soil and decaying matter and trees. And unlike neoformans, which is associated with pigeon drops, and there are 5 phenotypically indistinguishable species, which is a complex, which consists of Cryptococcus balicis sporus, Decagetii, Deuterogetii, Gatii, Sensu stricto, and Tetragatii. Okay, and then for the clinical reports, we only report as Cryptococcus gatii species complex. In terms of infection, it can be due to inhalation of the basidospore. Cryptococcus gatii primarily infects immunocompetent, but it also can infect immunocompromised, unlike Cryptococcus neoformans that primarily infects immunocompromised. And it can cause pneumonia and meningitis. And compared to neoformans, gatii is associated with high incidence of cryptococcomas, a collection of immune cell and organism. Okay, so this is the features about gatii. And in terms of diagnosis, cryptococcal antigen tests, where we have latex agglutination and lateral flow, and we can perform on serum and CSF. The clinical sensitivity is 90% to diagnose pneumonia, and the clinical sensitivity is 85 to 100% to diagnose meningitis. And based on CSF analysis, lateral flow perform better than agglutination compared to agglutination, which is 100% and 93%. So we move to case 2. So this is actually a case of urinary tract infection in Duchenne muscular dystrophy patient. So if you see the colony, it is actually a chalky, wrinkled white colony. And then you can see an arthroconidia. So this is actually an arthroconidia. So this organism is actually Trichosporan species. Okay, so in terms of Trichosporan, it is also a basidiomycetes yeast, and it is a component of Nomoflora. And there are 4 clinical isolates medically important, which is Trichosporum asahi and it can cause superficial infection of the hair shaft, where we call it as white pedra, and it can cause HIV and intravascular devices. And the treatment is basically voriconazole. Can you all hear me? Because, hello? Hello?
Yes, we can hear you,
Yeah,
Doctor.
okay. In terms of diagnosis of Trichosporum asahi, the colonies are white to cream colored. It is chalky, irregular folds, and the most important thing that we can appreciate the arthroconidia with the barrel shape, and the urine test is usually positive. It can be identified by API and MALDI-TOF. So in terms of the identification, Trichosporum is not a problem. Okay. And now we move to the third case. So this is a 66-year-old woman who is immunocompetent. She presented with recurrent erythematous lesion on her face and had been there for over 10 years of periods. Okay. So we can see that the colony is purple in nature. There is white at the periphery and purple or violet in color, and we can see a penicillium-like formation. Okay, but the phyllites is a little bit swollen and getting thinner above. So this is actually Purpurocilium lilacinum. Okay, so Purpurocilium lilacinum, the colony, we can have a violet colony. So it have a conidiophore. This is actually a conidiophore, long conidiophore. This conidiophore is supposed to be rough, which is not smooth. And then the phyllites are swollen. And then the conidia are ellipsoidal. Okay, so basically when we see a violet colony with a penicillus branching, or we call it as penyapu or broom, so this is a diagnostic of Purpurocilium lilacinum. So it is actually formerly known as Pacillomyces lilacinus. It is found in soil, and it has been used as a biological control agent against plant parasites, and it is considered as a opportunistic pathogen. It can cause infection to the eye, to the cutaneous and subcutaneous, and especially in immunocompromised. And this Purpurocilium lilacinum is intrinsically resistant to amphotericin B, fluconazole, and itraconazole. So this one is a very popular fungi in HIV patient. So I guess everyone gets this correct, okay? But I still got it wrong because of it can be because of the spelling, okay? So what is the term used for fungi which can grow at 25 and 37? And what is this fungi? So you can see that there is yellow to green, okay, yellow-green colony, and the most striking features is that it is pigmented. And again, you can see a penicillium branching, okay? So this is actually a dimorphic fungi, which is known as Thalaromyces malifii. Previously, it is under the family of Penicillium. So this is only one eye. So typically when you all send a specimen, especially from the blood, and we can see this type of colony with the red pigmented, we can usually guess that it is actually a Thalaromyces manifii. It is biverticillate, okay, and the penyapu is considered to be more broad. It can branch up to 5. Okay, so most of the time, when we have an HIV patient, we saw this bloom, we know that this must be a Thalaromyces manifii. So manifii is a dimorphic fungus in Southeast Asia, and it is actually a serious infection, especially in patients with HIV and AIDS. It is transmitted by inhalation, and it can cause infection to the skin, liver, spleen, and lung. And the treatment involve amphotericin B. Okay, case 5. So this is actually a bonus question because if some of you might see there is the answer in the picture B. Okay. So this is a case of COVID-19 patient with a multi-drug resistant fungal infection. So you can see that there is a swollen phylites. Okay. The colony can become black. Okay. And there is a swollen phylites, and you can see a conidia in mass. Okay. So this is a typical features of Lomentospora profilicans. Okay, so there is a flash-shaped phylites with ovoid conidia, and the conidia is actually in a group. Okay, so you can also have a colony which is full, black full, okay, which it is a rapid grower, and then can be downy. Downy is like a baldo, okay, so the conidia born in a small group, but the most important features that you need to remember that it has a flask-shaped phylites. So this is the most important features of the Lomentospora profilicans, and the conidia are aggregated in a slimy heads. Slimy heads meaning that it is in group. Okay, so Lomentospora profilicans formerly is known as Scedosporium profilicans, but after in the era of DNA identification, it is actually phylogenetically and morphologically distinguishable. We can differentiate between Lomentospora and Scedosporium. Okay, so it is highly resistant to antifungal, and the risk factor would include malignancy, cystic fibrosis, and solid organ transplant. And patient may have an infection with a pulmonary, bone, and joint infection. So again, you have to appreciate the phylites, which is special characteristic. Okay, so in the lab you might come across Scedosporium, other Scedosporium species like Scedosporium apiospermum. Okay, but you can see that in apiospermum usually it is singly or double, but the phylites compared the phylites, Scedosporium have a long phylites. Okay, and in Lomentospora it has a special phylites, and the conidia is in a slimy heads. Okay, now we move to the 6th case, which is dermatophytosis in a female. This female has a contact with cats. So if we are very familiar with 3 common species, is either MET or TEM, which is the Microsporium, Epidermophyton, and Trichophyton. Okay, and if you can see, there is a macroconidia, which is we can see the microconidia is quite distinctive, and then it have a thin wall, and the wall is rough, and this is actually, and the answer is actually Nanesia gypsia. So previously Nanissia gypsum is known as Microsporum gypsum, but now it has been renamed as Nanissia gypsum. So 3 common species for dermatophytes. We are familiar with Trichophyton, Microsporum, and Epidermophyton. Okay. So in terms of dermatophytes, there are many more dermatophytes like Iotroderma, Cytomyces, Epidermophyton, Lophophyton, Microsporum, Nanissia, and Trichophyton. And now there is also Guaromyces, okay? But usually, clinically, we will encounter these 3 species, which is Trichophyton, Epidermophyton, and Microsporum, okay? In terms of Nanissia gypsia, the colonies are usually flat, okay? This looks like powdery, okay, granular with deep cream. It has a pale cinnamon color. And if you look at the macroconidia, it is elliptical, but broad. And then you can see the cell wall nipis, and it has a very a rough cell wall. Okay, so this is how the Nanissia gypsia looks under the microscope. It can be easily identified by in the lab. Okay, so Nanissia gypsia is actually a geophilic fungus. It has a worldwide distribution, particularly in animals and human. And based on the multilocus phylogenetic study, it has been now Nanissia consists of 9 species. Okay, and it usually produce a single inflammatory skin or scalp lesion. Okay, so now we move to case number 7. So this patient, 25-year-old male, came to the dermatology department with complaint of nail dystrophy for 7 months. Okay, so we can see that that is quite a severe onychomycosis, and then the colony looks like green, and then it has a black dots, what we call it as it had a black dot, and then you can see a special features, which we call as perithecia or fruiting bodies, and it has a setae. It has a long filament lights, what we call as a setae. So this is how a Chaetomium globosum look like. Okay, so the colonies are growing rapidly also. It is cottony. It can be white in color, and it can become gray and olive in color later. And then the most striking thing about this Chaetomium globosum, it has a fruiting bodies, what we call as perithecia, which is big. Okay, it is usually black to brown. It can be in many shapes, and it has a hair-like brown to black appendages, what we call as setae on the surface. So it is not setan, it is setae. Okay, so this perithecia, it has an opening, what we call osteoles, and containing asci and ascospore. The spore for this Chaetonium is called ascospore. Okay, and then the ascospore can be one cell, but the diagnostic will be when we see the perithecia, which is the fruiting bodies. Okay. So again, how the Chaetomium can look like under the leptophenocotton blue. Okay. And we can see the black dots that we can call as ascocarps. Okay. So Chaetomium is actually a dermatiaceous, means black filamentous fungal. It is found in the soil, air, and plant. And it can become a contaminant, but also can become a human infection. It is the infection can be called as pheohyphomycosis, which is infection by the black fungus. This Chaetomium can cause fatal deep mycosis, which has one of the species, which is Chaetomium aetrobrunum, has been reported in immunocompromised host, and it can cause a brain abscess, peritonitis, cutaneous lesion, and onychomycosis. Okay, so whenever you come across this picture, remember of chaetomium. Okay, I believe some of us have encountered this chaetomium. Okay, so now we move to the 8th case, which is a young lady come with a cutaneous fungal infection. She has SLE. Okay, so from the picture we can see that the colony is white in color. Okay, it is actually usually cottony. Okay, and then the lactophenol cotton blue. The morphology reminds us a lady with a sanggol. Okay, so this is actually Saxinia species. Okay, so there are two species, which is Saxinia and Cunningham-Hamela. Cunningham-Hamela, they are not easy to sporulate, and we need to use water agar to make them sporulate. Okay, to see this beautiful formation. Okay, so Saxinia also fast growing, and then it has a sporangia. This is what we call as a sporangia, which is typically flash shape. But by our identification, we just think that this is a lady with a high hair, and typically a flash shape with a distinct spherical venter and long neck. It has a long neck. Okay, and darkly pigmented rhizoids. So dia ada akar. Okay, and the columella is prominent and diffuse. Sorry, this is actually the columella. Okay, and the sporangiospores. The spore for the Saxinia, we call as sporangiospore. It is small, oblong, and are discharged through the neck. Okay. Saxinia is characterized by the formation of flash-shaped sporangia with columella and simple darkly pigmented rhizoid. It is an emerging human pathogen that is mostly associated with cutaneous and subcutaneous lesion after stroma. And then Saxinia vasiformis was the only known species with worldwide distribution in association with soil, but somehow there are also identified species, which is Erythrospora, Doriceae, Lautrophoriformis, Oblongispora, and Trapezispora. Okay, to do the agar, to do the water agar, this is the slides that has been shared by Dr. Tan. It can be used by using Bacto agar with a distilled water. So we can produce the agar, and we can add the colony inside this water agar to see sporulation. But during my attachment in PGI Chandigarh, what they did is they did a very simple water agar. So they just take this petri dish, add the tap water, add the colony, and wait for the colony to grow and fish out the growth and do LPCB. Okay, so basically the simple technique by just using tap water is also can make this fungi to sporulate. Okay, this Saxinia. Okay. Okay, now we move to the case number 9. This is actually an autopsy case of disseminated fungal infection in immunocompetent patient. So this is also a family of Mycology. Okay, you can see there is a cottony colony, white cottony colony. So this is actually a Cunningham-Hamela bertholdtiae. Okay, so it has a special head features. Okay. So cunning hamela colonies, it grows very fast, white at first, but can becoming dark gray and powdery. And then the sporangiospore, okay? Sporangiospore are straight with verticillate or solitary branch. So it can branching as verticillate branching. So vesicle are subglobous. These are the vesicle, okay? The head are subglobous. And those sporangelia are globose. It can be varicose or short ecanulate, means that the permukaan of the spore is not smooth, and it is hyaline singly but brownish in mass. Okay, so this is how the head of the cunning hamela bertholatiae looks like. Okay, so sometimes you can see that it is either in brown. Okay, so these cunning hamela species are mainly soil fungi. Okay, and they are rarely isolated in a temperate region, and it has 7 species, and it is known to cause infection, often associated with trauma and immunosuppression. Okay, so those Succinia and cunning hamela are the same group with Rhizophus. Okay, so you have to appreciate that they have a broad hyphae compared to others. They have a broad hyphae. Okay, so we are now moving to case number 9. So this is actually a 26-year-old Hispanic woman. She presented with left lower extremity swelling, okay, with edema, erythema, and purulent drainage for 3 months duration. So this is a chronic skin problem, okay. So based on the picture, we can see there is a black colony, okay. So this is, and you can see even in the LPCB, there is hyaline and brown hyphae, and you can see there is a mass of conidia, which we call as a slimy head after the phylates, okay. So this is actually Phaeoacrimonium parasiticum, okay. So Phaeoacrimonium parasiticum, it is slow growing, okay, and the colony can become olivaceous gray to black with age, and then the hyphae, it can be hyaline to brown. So this is hyaline in blue to brown, okay, and the phylates thick wall. This picture is to show you it is thick wall, and it is not smooth. So you can see the phylates is not smooth, okay? And the conidia is in a slimy head or balls of conidia, and the conidia 4 usually is branch, okay? So in terms of species, there are Phaeoacrimonium parasiticum, rubrigenum, and so on. Okay, so usually patient also can have it can cause infection in immunocompromised setting, which is hematologic malignancy, in the setting of chemotherapy, stem cell transplant, rheumatoid arthritis treated with infliximab, and adult still disease managed with oral prednisolone. Okay, so for the antifungal regimen is not clearly defined, but it has been a good outcome with itraconazole, amphotericin B, voriconazole, terbinafine, and surgical debridement. Okay, so in the lab, usually we will see a white, initially a glabrous white colony that we change after 2 weeks or 3 weeks. Okay, and then we can see that it is either give a phylates, the conidia 4, it can branch and give a phylates. Okay, so this is how we can encounter the types of colony in Phaeoacrimonium parasiticum. And in our practice, we can confuse this Phaeoacrimonium with an acrimonium. because sometimes the colony did not change. Okay, so but it share the same features. So acremonium, it have an all shapes, same. All shape means that it is broad from the base and becoming smaller, and then it has a slimy balls of conidia. Okay, so in terms of phaeoacremonium, you have to see that the hyphae becomes brown. So this is how we differentiate between acremonium and phaeoacremonium. Okay, so we move to the 11th case, which is a case of subcutaneous mycosis in a pediatric patient. And we can see that there is a white colony and there is a small colony at the peripheral, which we call as a satellite colony. Okay, and we can see a very prominent spore, but and then this is actually a Conidiobolus coronatus. Okay, it has a flat white to cream colored glabrous satellite colony. This is what we call as a satellite colony. So previously, this Conidiobolus coronatus is under the zygomycete, and then it has becomes a member of Entomophoralis. Okay, so it is not under Mycology, previously known as zygomycete. It has become another member. Okay, so the colonies of Conidiobolus grow rapidly, and then it later become a fine powdery with a satellite colony. Okay, so this is a very distinctive features of the Conidiobolus coronatus. Okay, so the lip of Petridis soon become covered with conidia because they are forcibly discharged by the conidiophores. And the color of the colony may become tend to brown with age. And then about the conidia, okay, the conidia has a special features which we call as hair-like appendages, which is called also as a villi. Okay, so this conidia, it can germinate either to produce a single or multiple hyphae tubes. Okay, and which we call as papillae. Okay, this is actually we call as a papillae. And it also can replicate by producing multiple short conidiophore. So it will become elongate and produce a hyphae conidiophore. Okay, so this is a typical features of Conidiobolus coronatus. Okay, so Conidiobolus coronatus is commonly present in the soil and decaying leaf. It has a worldwide distribution, and especially in the tropical rainforest of Africa. So human infections are usually restricted to the rhinofacial area, and there are reports on disseminated infections in immunocompromised patients. Okay, so whenever you saw a distinctive conidia, what do we call this now? Okay, conidia. It has a papillae. Okay, so this will give you the identification of Conidiobolus coronatus with the satellite lesion, satellite colony. Okay, but you have to differentiate Conidiobolus from the Basidiobolus. Medically important Basidiobolus would be Basidiobolus renarum. Okay, so this is actually an image of Basidiobolus renarum. It also can have a satellite colony, but it shows cinnamon-like color, and it have a big zygospore. We call it as a big zygospore, conidia, and a sporospore. The spore is called sporospore. A distinct swollen area just before the conidium. So if the Conidiobolus have a papillae, this Basidiobolus have a distinct swollen area below the conidium. Conidia and a sporospore. Okay, so you have to differentiate these both because sometimes in the lab we can see it as the same, even though because in the atlas, of course, the differences would be vary, but once you see in the under the LPCB, it can become the same. Okay. Okay. Now we move to the case number 12, which is a cutaneous fungal infection in a delivery personnel. So it is a chronic fungal skin infection in this man. What is the type of the candidiasis, and what is based on the picture on the orange color. So this is actually a Sporobolomyces salmonicolor. Okay, so this yeast, it produces a ballistoconidia. Okay, so Sporobolomyces salmonicolor is actually a Basidiomycetes yeast as well, and it is pigmented due to the carotenoid production. It may look like Rhodotorula, so usually we will confuse it with Rhodotorula. It can be found in air, tree leaf, and orange peel. And it is common as the Sporobolomyces salmonicolor is commonly associated with human infection, in particularly in immunocompromised patients as well. And it can cause a lymphadenitis, cerebral infection, fungemia, and dermatitis. Okay, so the things about it producing a ballistoconidia, we can do a test in the lab where we put a colony in one plate, and then we put another plate above that colony, and we tape together to see this mirror image. Okay, so about the colony, it is slightly rough with a characteristic of salmon pink, and we can see the satellite colony because when it produces ballistoconidia, it will the colony will shoot up this ballistoconidia and it will land as another satellite colony. Okay, so it is actually urease positive. Okay. Okay, now we move to case number 13. This is actually a CDC report in October 2012 when there is an outbreak of fungal meningitis associated with methylprednisolone injection. So we can see there is a black colony, okay? And we can see a very distinctive features of a macroconidia of Azerohilum rostratum. This is actually a black mole, okay? So the colonies are gray to black, okay? And then it can be become olivaceous black in reverse, and the conidia are straight, curved, and then it is actually ellipsoidal to fusiform. But the most important features of this Azerohilum conidia is that it has a strongly protruding truncate hilum. So this is actually a strongly protruding truncate hilum with a septum above. Okay. So it is also have a distal septa. It doesn't have a true septum, it have a distal septa. Okay, because of the cells inside the conidia. So whenever you see a conidia with a strongly protruding truncate hilum with a hilum and a septum, so this is a diagnostic features of Azerohilum rostratum. Okay, so it can be found on plants and soil, and it can cause infection by contaminated injection, like the outbreak, eye trauma, or skin injuries. And it can cause fatal fungal meningitis, brain abscesses, sinusitis, keratitis, and skin lesion. It is a rare opportunistic pathogen, especially in immunocompromised individual. Okay, so we have another 4 cases to go. So this is a case of Tinea nigra, or we call as black pedra, on a palm of a young gentleman. Okay, we can see the black colony, and we can see a special features of the conidia, which has a black septum. Okay, so this is actually Horte wernickeii. Okay, so Horte conidia, so it produces a brown to dark olivaceous, it produces a black colony. Okay, but the most important thing is that it is a 2-cell conidia. It can be 1 to 2 cell, and it has a septum. Okay, so whenever you have a colony with a striking features of a septum, so this might be Horte wernickeii. Okay, so if we do the KOH from the sample, we can see a hyphae, okay, black hyphae with a black colony. When once you do the LPCB, you can see a very distinctive features of the conidia with a black septum. So we can easily diagnose it as Horte wernickeii. Okay. For the Whitepedia, it can be caused by just now Trichosporan species. Okay. So the Trichosporan species will have these features. Okay. I just show for the interest. Okay. Okay, case number 15. This is a case of a 78-year-old woman with comorbidities, which include hypothyroidism, bronchiectasis, type 2 DM, atrial flutter, renal impairment, and chronic back pain. Okay, so actually this woman has a very complicated medical issues presented by the journals. Okay, so based on the picture, okay, we can see a glabrous colony, okay, which is like a yeast-like colony. Okay, but once we did an LPCB, it will have special features cells that we can call as morula. So this is actually an organism called Prototica wickerhamii. So I got this Prototica wickerhamii with my other colleagues during our master exam. Okay, so Prototica wickerhamii is actually not a fungi. It is not under the kingdom of fungi. It is actually an algae. It is found in environment, including soil. So it is actually in the kingdom of Protista. And it is a chlorophyllous algae. Okay, but it can cause human infection, human prototicosis, which can range from a cutaneous lesion to systemic infection, especially in immunocompromised individual. And another species will be Prototica ciferri, formerly known as Prototica zofi, which is also associated with human infection. So you can see it can be called as a sporangia. Okay, this is a special features of the Prototica wickerhamii. So in the lab, we can see a yeast-like colony, okay, on the SDA. We have a morula-like sporangia, and it is usually can be identified by our API identification yeast and by MALDI as well. Okay, so this is how bad human prototicosis can look like. This is an infection to the upper limb and left lower limb of the patient. Okay. So second last case is a case of skin ulcer in a diabetic man with a liver cirrhosis. He contented with a swarm water. Okay, so in this cacoflow white, we can see a ribbon light. If we do LPCB also, we can see a ribbon light. Okay, so in the lab, when we want to differentiate whether this is a Mycology or not, we have to see there is a ribbon-like hyphae to show that it is under the Mycorrhizae or zygomycetes. Okay, the term zygomycetes is not being used now, but still, because the clinical, we will say it is either zygomycosis. So the term still exists. Okay, so you can see that the colony is not like a fungal. Okay, it's not like a mole. Okay. But we see a ribbon-like. Okay, so in Mycorrhizae, we will see a cottony-like colony. Okay, so this is actually a Phytium species. A Phytium species can give you a flat colony and a ribbon-like hyphae. Okay, in LPCB. So whenever we have a patient with history of severe infection, okay, patient can come with a severe swelling with multiple ulcers, okay, and we can see inflammatory lesion and abscesses in the MRI. So this is a typical severe infection by the Phytium species. Okay, so Phytium, like Prototica, is not a fungi. It is in a kingdom of Straminophila, okay, in the class of Omycetes, okay, and it is actually an aquatic organism found in water, moist soil, and decaying matters, and Phytium insidiosums, okay, usually causing phytiosis in humans, which can lead to chronic granulomatous infection, often involving the skin, cornea, and GIT. Okay, so this is how a Phytium colony can look like. And it usually have a flat, flathery colonies with submerged growth on the SDA. So we can see aseptic hyaline hyphae. Okay, and it have a zoospores. But so phytiosis is rare. Okay, it has been reported mainly in Thailand, but I see also in Malaysia there are few reportable cases, and there is actually no standard therapy, but people still use amphotericin B and azo for the treatment. Okay, so this is how a Phytium insidiosum look like. Flat colony, you see a broad hyphae. You have to think of Phytium insidiosum. Okay, so now we come to the last slide. Okay, so I just choose this case because I think it is rare. It is nice to know, and then it looks like a parasite. Okay, so this is a case of 73-year-old male gardener. He presented with a conjunctival injection in the cornea of the right eye. So so we can see the cornea. The cornea, it has a white to green material. Okay, injected cornea, and the colony is white in color. But when we do LPCB, we can have this parasite look like. It has a median cell body, which is 3 cells and 3 appendages. Okay, so this is actually a Pestalothiopsis species. Okay, so in this case, this is caused by Pestalothiopsis clavispora. Okay, so in the LPCB, we can see the conidia with 3 apical appendages. Ini tak nampak sangat lah, tapi you can see it looks like a parasite. So this is actually a fungus. You have to believe this is a fungus. Okay, this patient is actually have been given micafungin, but it can relapse after 8 months. So they give topical micafungin to the patient. Okay, and the Pestalothiopsis, it is rare. Okay, it has a median cell and a basal cell, basal appendages, and it has apical appendages. Usually it have a three apical appendages. Okay, so it is a large diverse genus of fungi. It is actually a plant pathogen, and it live in the plant as well and act as a decomposer. Okay, So that will conclude the discussion on the quiz itself. Thank you.
Thank you, Dr. Siti Hawa. Okay. We have one question for you in the Q&A box by an anonymous attendee. Can you share your experience on using MALDI-TOF for identification of molds?
Oh, actually, to be frank, I don't have many experience identifying mold by MALDI-TOF. Okay, we are still what is the words? We are still in the process to motivate all the labs in Malaysia to use MALDI-TOF. But of course, I have a feedback that it is not easy to identify mold by MALDI-TOF. Usually, we don't get the identification, but still, it is a good platform to identify yeast, and we are using it for the correct identification. But somehow you will have problems to identify a colony which is quite, how do I call it? Like a Cryptococcus colony. How do we describe it? And so we have to make sure that we get rid of the apa? How do I describe it? It has a thick wall. The colony is hari ni semua perkataan hilang ni. The mycoid colony kan. So you have to remove that mycoid colony and you have to take the colony inside and to do the MALDI so that you will not have problems in identifying. But in terms of mold, we are still encouraging our labs in the government hospital to use because the thing is that it is not we say that it is not really established yet. Okay. Or any other participants who like to comment on that? Who have been using MALDI-TOF? Like Johor?
Okay. We have two people with their hands raised. Aziani, I will allow you to talk and ask you to unmute so you can ask your question. You can unmute your mic. Okay, in the meantime, there's another person, iPhone. We will also allow you to talk and ask you to unmute. If you have any questions, please ask them.
I usually said, ask me no question and I'll tell you no lies.
Project the.
Okay, thank you, Dr. Siti Hawa. Okay, I will now project the QR code as requested. As requested.
I also would like to thank everyone for the participation. Thanks all the participants, the moderators, and all the presenters. Thank you for making this seminar successful.
Thank you, Doctor. Okay, so this is the, I hope you all can see the QR code for today's attendance and also the feedback form. So we'll give you a few minutes to scan if you haven't done so. We have also shared back the link in the chat box. Okay. And for our channel,
So all recordings and slides.
Ada adik juga lah itu Hana. Ada juga.
Okay. All right. Yeah. Okay. Our slides and recordings will be uploaded to the social media channels of MSIDC MMIG at X, Instagram, as well as on YouTube for your reference. Please take note. Right. Okay, with that, we officially conclude this year's Fungal Awareness Week. On behalf of MSIDC Mycology Interest Group, we thank our speakers and participants as well as our IT team for your time and contributions. We hope to see you again in our future activities. Thank you so much.
Thank you and have a nice day.